I don't know how these got out of sync, but this, I think yours is probably in this order. Because the first one that you have is this one. Yeah, I don't know what happened. I don't know how this one got into slide 7 and got saved. But this chapter is metabolism. So by definition, metabolism, all the chemical reactions that happen in living systems, that's metabolism. And that gets split into sort of 2 different types. You either have catabolism or you have anabolism. And these 2 together are really important because one is the building reactions, the other is the breakdown reactions. So catabolism is the breaking down. So where you take large molecules and you break them down into smaller parts. So when we talked about chemical reactions, what kind of chemical reaction would that be? Or we're identifying types of chemical reactions. If you go from one thing and you have lots of products, that is? So in the 4 different types of chemical reactions, we had single displacement, double displacement, synthesis, and decomposition. What would you call this? This is decomposition. So these are decomposition reactions, digestion, energy production. In fact, catabolism is what produces energy. And it does this by breaking chemical bonds. So we started off with the 4 little circles linked together and end up with 4 single circles not linked. When you break chemical bonds, you release energy. So that is going to provide the little building blocks in order to do anabolism. So anabolism will be taking little individual units and linking them together into larger structures. So what type of reaction would that be? It's considered a synthesis. So synthesis reactions are when you build things up. You have lots of reactants and fewer products. This is your energy demanding processes. So this uses energy. I always think of catabolism and catastrophe, like everything falls apart. Okay, so catabolism, everything falls apart. Decomposition, example. Anabolism, I always think of anabolic steroids. Because people that take anabolic steroids like build muscle mass. Okay, so they build muscle, and so those are your building reactions. Those are the really energy demanding types of reactions. So in looking at them, they're really sort of one feeds the other. Doing catabolism, doing breakdown reactions, provides building blocks and energy. So notice in this reaction we get ATP. In this reaction we're going to get the small components that your cell will need in order to build larger structures. And then anabolism is the energy using and the building block using reactions to be able to link molecules into large structures, to build structures like cell membranes, to build enzymes, to build proteins, to be able to increase the size of the cell so the cell can go through division, do protein synthesis. All of those are anabolism. All of those, whenever you think of trying to build something up, that's going to be the anabolism reactions. So ATP, which I have listed down at the bottom, it's called adenosine triphosphate. ATP is useful because it provides uniform energy. Some chemical reactions produce a lot of energy when they break bonds. Other chemical reactions only produce a little. And so you need to make sure that the cell has a uniform energy source, kind of like a battery. So like you use a AA battery, you don't use a AAA battery, you don't use a D-size battery. You use the energy that's applied in a certain battery because that's a universal energy source. And if you hook that equipment up to another battery, it may end up sort of like creating a surge, like a power surge. Too much energy, that would end up affecting it. Same thing happens with the cell. We want uniform, consistent energy for the cell, and it is somewhat storable. So we can store this energy in the cell, so we don't necessarily have to use it right now. So we can take and break down molecules and trap that energy as ATP, and then when the cell needs to build molecules, you have that ATP sitting right there. So the where do these things take place? Inside of the cell, and this is the little generic cell. Energy production really happens in one of two places. It happens in the cytoplasm or it happens in the mitochondria. The mitochondria have the names of being the powerhouses. So they're the powerhouses of the cell. You'll see that they are actually where the bulk of your ATP is produced inside of the cell to be able to power all of the building reactions that go on. Now, when you look at a mitochondria up close, you see that it's actually a double-membraned organelle. The outer membrane's really like a cover. The inner membrane's this really ruffled-looking layer. So there's sort of like an inside, and then there's an inner in the middle, and then there's outside of the mitochondria. So you have a couple of different types of spaces that are produced. So we'll talk a little bit about enzymes that are found in that inner ruffled layer. Okay, so now this breakdown part, which was supposed to be up here. First one, carbohydrates. So in this one slide, I have polysaccharides and I have simple sugars. The first one is talking about the polysaccharides. So this would be like starch. Remember that starch is hundreds to thousands of glucose units. Really large molecules, size-wise, the only thing bigger is DNA, okay? Polysaccharides are like the second largest of the nutrient groups, molecules found. So in order to be able to get any nutrients from these polysaccharides, they have to be digested. So we're talking you have to break hundreds to thousands of glycosidic bonds. So each one of these little links, you're going to have to break. So if we have a chain of 1,000 of them, you've got to break 999 little glycosidic bonds. That takes time. This is really like, takes the longest of any of the nutrients. So if you think about digestion, you ingest things, it goes in your mouth, you chew it up, you mix it with saliva. Saliva actually starts this breakdown, but then it passes along, goes down the esophagus, hangs out in your stomach for a while. Not a whole lot of sugar digestion or polysaccharide digestion happens there. It really kicks off once it gets into the small intestine. So the small intestine is this 20-foot tube, and it takes about 2/3 of the way through the small intestine before you really start absorbing this. So time-wise, you're talking about a couple of hours. 2 to 4 hours of this processing and digestion that has to go on with starches before you're going to get molecules this small. And they have to be that small. They can't even be disaccharides. So oligosaccharides, any more than 5 sugars, maltose, you don't absorb it. It has to be all the way to the point of being simple sugars, just glucose molecules before it gets absorbed. So this is the longest processing time of any of the nutrients. But these, what are these ones? They're disaccharides. Disaccharides are part of the simple sugars, but even disaccharides are too big to be absorbed. So you look at them, you're like, well, there's only 2 sugars, but that's actually too big. You have to break them down into single sugars. But how many links do you have to break?
2.
Well, really just 1. You just got to break that disaccharide in half so that you end up with 2 little sugars. So only 1 bond needs to be broken. Unlike your starches where you're talking about hundreds or thousands of bonds that have to be broken to get it all the way to the glucose size. So maltose gets acted upon by an enzyme, same with lactase, same with sucrase. This happens early in the small intestine. So these sugars don't hang out way down the small intestine. They come in the small intestine, they only have to be broken once and they get absorbed. So you're going to see that the disaccharides get absorbed, broken down and absorbed in the first couple feet of the small intestine. Because once they get monosaccharide size, they can get absorbed. So they're absorbed quickly, especially compared to their large polysaccharide counterparts. So these, glucose, galactose, fructose, what are those? They're all mono. What kind of processing do they have to have? None. They're already their smallest piece. They're only single units. So they're small enough to be absorbed. There's no processing, no digestion that has to happen. They're already small enough. They can get absorbed through the lining. Glucose can get absorbed through the lining in your mouth. Galactose and fructose get absorbed early on in the lining of the small intestine because they're really like the breakdown products of disaccharides. So these are the fastest. They're the fastest to get absorbed because there's no processing. So when thinking about the carbohydrates, polysaccharide is going to take you a couple hours to actually break it down completely and absorb it. Disaccharides pretty quick as soon as it gets into the small intestine. Monosaccharides, though, are going to get absorbed the fastest because there's really no processing that has to go on. We're going to talk about glucose and how the cells use glucose for energy, but what about proteins? So proteins are chains of what? Amino acids. So chains of amino acids. So kind of like your polysaccharides, you can't absorb these until you break the peptide bond. So like polysaccharides take a couple of hours to break them down, digestion starts in the stomach. That is why stomach acid and pepsin, an enzyme secreted into the stomach, that's how they go into action. But notice it's not until you get into the small intestine, until you start getting single amino acids like glucose, you've got to break proteins all the way down to single amino acid size before they can be absorbed. So a couple of hours. This is, again, kind of a 2 to 4 hour processing. Enzymes break them down. Most of your absorption happens middle and late of the small intestine, kind of like the polysaccharides, because they're pretty close to size of a polysaccharide. Proteins anywhere from 50 amino acids, which would be a little protein, that would get broken down faster than starch would. But you can also have ones that are 250, 500 amino acids in the long chain. So that would take quite a bit of breakdown. Large proteins would take a while. The last one, the fats. Lipids, which are your fats and oils. They exist as triglycerides. Triglycerides, shown at the top, are just long chains of carbon, 3 of them linked together. So that long chain of carbon, that's the one that they look like this little long wiggly tail, that is the fatty acid. So a triglyceride is just 3 of them linked together. So this can't be absorbed. It has to get broken down first. This again happens in the small intestine. The pancreas helps with this, and it secretes an enzyme that helps to break the triglyceride into individual fatty acid units. Those are small enough to be absorbed, but there's a problem. What happens when you put fats and oils in water?
They don't mix.
They don't mix, they separate. So fats and oils wouldn't mix. If they got absorbed into the bloodstream, they would end up forming like little oily pockets in the bloodstream. So this is where you have bile. So bile is a lot like soap. So remember that we said that soap makes those micelles. Soap makes those little spheres that pull the nonpolar molecules into the center. So you use soap because it helps to get greasy, waxy, oily type of material off your hands. So the micelle pulls the oily material in. So that's what bile does. Bile is secreted by the liver, and it does what's called emulsification. So it emulsifies the fatty acids. It coats the fatty acids, and it makes them soluble in water. And then they can get absorbed into the blood. This is why people that don't have a gallbladder can have problems with eating too much fat, is because there's not enough bile secreted. So the gallbladder stores that bile, but if there's not enough bile secreted, then that fat will not get absorbed. It'll just pass straight through the GI tract. So people pretty quickly learn without a gallbladder what limitations they have in terms of fat intake, because you just ended up with just a holy mess, like you have the holy diarrhea mess. And that's just because of lack of bile. If you didn't have bile, it's not going to emulsify the fat and you're not going to absorb it. It's just going to pass straight through you. Once it gets absorbed, it actually has to have a carrier. So there is a carrier that also gets added in that kind of looks like a micelle. So the fat's in the center and the little carrier covers. Remember we talked about cholesterol? Cholesterol also has to have that carrier, the HDL, the LDL carriers. The fatty acids travel along through the bloodstream, having a carrier to keep them dissolved, because otherwise they would separate out. Oil and water, and your blood is totally water-based, so oil and water won't mix, so you always have to have the chylomicrons, or the early carriers, but then later on it gets packaged into these lipid carriers. So not only do you have to break triglycerides down, you've also got to have some way of getting it to be absorbed, and that's the job of bile. Okay, so once all of these things are absorbed, once the proteins are broken down into the amino acids, they can get absorbed. Once the sugars are broken all the way down to the monosaccharide size, they can get absorbed. Once your lipids are broken down to the fatty acid size, so just the single chains, they can get absorbed. So those are kind of like the 3 things that get absorbed as nutrients. The cell can use all 3 of these for energy. The one that we are going to talk about is glucose. So we're going to talk about the center one, and then at the end we'll talk about how you can actually use amino acids for energy, how you can use fats for energy, and how they differ just a little bit. So at the very end, at the very end of that page, I have a couple of what I call metabolically important or metabolically active molecules that are involved. So the first one we said is ATP. The job of ATP is to trap energy when you break chemical bonds. It's nice and uniform, it's storable, so those are all the benefits of ATP. But now those other ones, they're going to come into play, NAD and FAD, they are going to come into play in the dismantling of glucose. So glucose is C6H12O6. So we're going to take glucose and we're going to combine it with oxygen and we're going to make carbon dioxide and water. So can you see that this is decomposition? We start with 7 molecules, we end up with 12. Okay, looking at that balanced equation, we started off with 1 glucose and 6 oxygens. We're going to end up with 6 carbon dioxides. So that means we had that chain of 6 carbons making the sugar. We're eventually going to break every single carbon apart. So there'll be individual carbon dioxide molecules. We're also going to take these hydrogens and we're going to use them to form water. So we're going to have to pull hydrogens off to try and make this glucose smaller and smaller and smaller, breaking bonds and generating energy as we go. So that's more or less like the steps that are going to come into play. So there's ATP up at the top. Here's NAD and NADH. So really, this is how it starts off, and then it does this. So NAD, you don't have to remember the name of it. It's called nicotinamide adenine dinucleotide. That's where the NAD comes from. And it's actually made from a vitamin. So niacin is vitamin B3. So it's one of the parts of that NAD molecule. And we'll talk about, there's a couple of other B vitamins that come into play in this. But what NAD does is it picks up a hydrogen. The hydrogen comes off of glucose. So it's one of the hydrogens in the glucose molecule. That is the source of the hydrogen. As we begin to dismantle the hydrogen from glucose, NAD can pick up 1. FAD can pick up 2. So if you see the 2 reds, see the 2 red hydrogens, and this is really like, should go that direction. So over on the right side is FAD. Over on the left side is FADH2. So that's an FAD molecule that's picked up 2 hydrogens off of glucose in the dismantling process that we're going to do. So this one stands for flavine adenine dinucleotide, and it's actually another B vitamin. So it's riboflavin. So riboflavin is B2. Niacin's B3. So 2 B vitamins important in metabolism. They're also water-soluble vitamins, so these are ones that you need regularly in your diet. But you get them from grains and vegetables, like in plenty. You don't really ever worry about running out of them unless you're malnourished. You end up with problems like it. But under general conditions, most people just in the food that they eat every day, it's plenty. All right, so then the last one. So in this, for NAD and for FAD, I would just say that they're the hydrogen carriers. They pull hydrogen off of glucose as it's being broken down. The last one in there is called coenzyme A. Coenzyme A, another B vitamin. Coenzyme A is made with pantothenic acid, which is B5. So 3 different B vitamins all involved in this processing. The job of this, and we just call it CoA. So it's coenzyme A, as it's a really long word, but I just call it CoA. Its job is to pick off a 2-carbon group. So glucose is made of 6 carbons. In the processing, CoA is going to clip 2 carbons off of our glucose molecule. And that's what we can use to further break down and make carbon dioxide. So this 2-carbon unit comes from glucose. So we'll see how that gets pulled off as we go. But that's these sort of helpers. They just kind of hold the molecules or hold the piece of glucose for a short period of time in the energy production step. Okay, so on the back page, what I did in the back page is I split it into the 3 steps of energy production. So as we go through glycolysis, citric acid cycle, and then electron transport, you can just add any notes, underline anything that you see as being significant, but I kind of already filled them out just so that you can kind of follow along instead of worrying about writing every single thing down. Okay, so glycolysis. So glycolysis is the process that's used to describe how you take glucose and split it in half. That's really all that happens. And it takes 10 reactions. And you do not have to memorize the reactions, okay? But it's 10 actual chemical reactions have to take place. And we're going to take one 6-carbon glucose molecule, and we're going to split it into 2 3-carbon molecules. So we're just splitting it right down the middle, directly in half. The first part of it, and this actually shows reactions 1, 2, 3, 4, 5. So see up here, can you see how this kind of looks like a glucose? So these would be carbons. So it looks kind of like the ring shape of glucose. So in this, see at the very bottom, can you see that there's 2 3 carbons, so both of the 3 gray little balls, they've been split in half. So what you end up with is 2 3-carbon molecules. So we said we're going to make ATP in this. But what are we doing in this step? In this first 5 steps? Are we making ATP? What are you doing? What happens to ATP in step 1 and step 3? It's actually getting broken down. Like, so ATP is becoming ADP. So when I first listened, I was like, this is like exactly opposite of what we're saying. So the goal is to make ATP, but they call this the energy investing phase. So you actually have to use 2 ATP to start the process. And the reason you need to is you see where those 2 yellow phosphates are? Those phosphates get added 1 to either side, and that weakens the bond in the middle. So it allows it to break. So glucose doesn't just randomly break on its own, but by adding a phosphate, that allows for bond rearrangements to occur. Second phosphate gets added, and then you can split it. So that'll split it into 2 separate molecules. So we use 2, but in steps 6 through 10, how many ATPs do we make? You make it step 7, you make 2, and then in step 10 you make another 2. So if we make 4, but we had to use 2, you still come out ahead. Okay? So we had 2, so 4 ATP are made or generated in glycolysis, but we had to use 2 ATP to get it to start. So that means 2 ATP are produced in glycolysis. What's another one that you see up at the very top? We make some NADH. So that's a couple of hydrogens get pulled off of the the glucose skeleton that's left behind. So we get 2 ATP and we get 2 NADHs. This all happens in the cytoplasm. This requires no oxygen. They call this an anaerobic step. So you can actually do this without having oxygen. So this is an anaerobic process. You don't see O2 used anywhere in here. But for us, If we have oxygen, then we can do this, continue the process aerobically. So the word aerobic means with oxygen. If we're doing this without oxygen, then you're going to have what they call anaerobic. So anaerobic means no oxygen, aerobic means with oxygen. If you do anaerobic glycolysis, your final product is lactic acid. So it's lactate, which then develops or creates or generates lactic acid. So what do you know about lactic acid? What does it do? No, well, that's lactose, and then there is it's not the same, it's a similar molecule. It's not exactly the same, but when do you make lactic acid? Your muscles. Okay, so when you're working out, if you work out under very high exertion, then your muscle cells go anaerobic. They can still use glycolysis to make energy, but they're going to start making lactic acid. And how does that leave your muscles feeling? Sore, tired, okay? So when this builds up in your muscle, that is going to lead to that soreness. So when do you do exercise that's so high stress that you can't get enough oxygen in? What would be an example?
Run a marathon.
Well, most marathon, if I ran a marathon, yes. I would never get there, maybe days later. But think about something that's more, because anaerobic is meant to be short-term, high force. So what's an example of that? Okay, lifting weights, sprinting, okay, doing something like the 50-yard dash, where you run the half length of the soccer field just as fast as you can. And so do you breathe while you're doing that? No, you're just going as fast as you can. That ends up being anaerobic. So sprinters, weightlifters, those blasted Ninja Warrior people. So with that, and I can tell you because you don't know her, but I have a really good friend that started doing the P90X, which is kind of like what is the gym? It's CrossFit. She started doing those, and so she does those crazy burpees where you jump down to the ground and you jump back up, and she does these deep knee bends with kettlebells, these 5 and 10 foot weights. She jumps up on boxes that are this high, freestanding jumps. Okay, she just does all of these things, and I'm okay. So we first started doing. She's been doing this for 3 years, and now she has no body fat, and she's a muscle. I'm you look like a Jack Russell. Okay, so she's just like that. But she first started, she could not roll over in bed. When she first, the very first week she did this, so she went 4 days a week. She did this day after she did her very first session. She was nope. Her husband had to roll her over. She was I cannot even pull myself up. So he was sitting her up, and she was oh dear. She could hardly raise her arms. That's because of extreme lactic acid. Because she's doing really, she only does these her class only lasts 40 minutes. That's it. Okay? But in that time, they do very high impact, very high exertion types of exercise, and so it's a very short term. So her body has now adjusted to this anaerobic processing. So now when lactic acid builds up, it doesn't bother her muscles as much. And that's why bodybuilders, so they can lift these extreme amounts of weight, and it really does create a lot of lactic acid buildup, but you kind of adapt to it over time. But initially it was really just entertaining just to be look over there. She's what? She can hardly move, but that's because of that. So anaerobic is those high force types of exercise. So if you do that with your skeletal muscle, then it's going to build up lactic acid. It's going to make you sore, and it's even going to make you weaker. So the next day, you can't lift your arms hardly at all. So short amounts, only 2 ATP, but that's enough to survive. right? So if something's chasing you, that 2 ATP per glucose, that may actually be a survival mechanism for you. Yeast, they have actually evolved so that they do this all the time. They like to live under these low oxygen conditions because they can generate ethanol. So they do a very similar process to glycolysis, but at the end, they don't stop at just breaking their glucose in half. They convert the two products of glycolysis into ethanol. And so when ethanol is present, guess what can't grow there? Bacteria. So yeast have actually evolved this ability to make ethanol and live in low ethanol conditions, and other microbes can't. So it's really like allowed them to flourish. So because they're just little single-celled organisms trying to get along. But if there's bacteria around, the bacteria are going to eat all their nutrients. So they just make a little ethanol and all the bacteria die off, and then they're like golden. So then they just hang out and stay there for long periods of time. So that's really, and that was actually discovered all the way back in ancient Egypt. They actually found evidence that there was fermentation, that they made beer and they made wine and stuff all the way back thousands of years BC. So it was something that once it was discovered, everybody else thought it was a pretty good idea too. So here's this one. So if we take glucose, we're going to make 2 of these. The 2 3-carbon units are called pyruvate. Okay, so if you count 1, 2, 3 across, this is the pyruvate. There's 2 of them. We got 2 ATPs and we got 2 NADHs in this process. That's glycolysis. Happens in the cytoplasm. It's considered anaerobic. Now, if you stay anaerobic, you're going to make lactic acid. So lactic acid is the buildup that creates the soreness. If you were a yeast, then you would make ethanol and live happily. But now if you keep breathing, so if you go out and you do, like marathoners train and they want to stay aerobic, okay? Aerobic means they keep air, so they make sure oxygen is present and available. So if oxygen is available, we're not going to stop with glycolysis. We're going to go on to the second step. So the second step, you have to take the byproducts, the pyruvate, and you have to take them into the mitochondria. Because remember I told you the mitochondria are the big powerhouses? So we're going to take pyruvate and we're going to move it in. And in order to move it in, we're going to clip off carbon dioxide. See the blue, like kind of the blue boxed area of pyruvate? Can you see how clipping that 1 carbon off makes carbon dioxide? And it leaves this 2-carbon group that the coenzyme CoA, is going to pick up. And this can now carry it into the mitochondria. So pyruvate can't just go into the mitochondria on its own. It has CoA as its carrier. So that one that I told you up on the front, that coenzyme A, it's going to carry these products from pyruvate, drop off a carbon dioxide, but take the other 2 carbons in. So in the mitochondria, the next step is what's called the citric acid cycle. And I'll tell you, if you've ever heard of these, the TCA cycle and Krebs cycle. All the same cycle. Because I've had some people be like, I don't really know what this one is versus that one. They're exactly the same. So Dr. Krebs was the one that first defined it. So they called it after him. But then because citric acid, the citrate that's made here, so the citric acid cycle came about as a terminology to call it. So any of those, exact same thing. It's just like 3 different ways of writing the same thing. So here, I don't expect you to remember any of these structures. I don't expect you to remember any of the names. All we're really going to look at is like what happens. Okay? This is a series of 8 reactions. The 8th reaction reforms what's going to be used to do reaction 1. So that is why they say it's a cycle or a series. So it goes in a circle. It takes 2 turns of the cycle per glucose. Because remember, you had glucose, you split it in half. Now you got these 2 pyruvates. So both pyruvates go into the mitochondria, carbon dioxide gets clipped off. So we're going to run this cycle twice for 1 glucose. And really all that happens in these is we have bond rearrangements that occur. We rearrange bonds using enzymes to try and group together carbon and 2 oxygens and clip off carbon dioxides. Because remember, when you break bonds, you release energy. So we're trying to clip them off so that we can release that energy. So here we have a 4 carbon group. Here comes the 2. So now I've got 6. All that I'm doing from this one to this one is I'm really just moving an alcohol group from carbon 2 down to carbon 3. So like I said, it's really just rearranging bonds. That's all you have to remember. But step 3, I make NAD into NADH. So I make an NADH, and I'm going to do this cycle twice. So in step 3, I'm actually going to make 2 NADHs, and I also make carbon dioxide. So the little blue here, or it's actually this one over here, that CO2 gets clipped off. Step 4 is where I get the other carbon dioxide. So remember, I got 2 carbon dioxides came off when the CoA came into the mitochondria. Here's where the other ones are. So that's really where all my carbons go. Your carbons of glucose become carbon dioxide. That allows me to pick up more hydrogens off of the remaining bits of the glucose. The only place I make ATP is right here. I only make 1 ATP per turn. So it means I make 2 of them. Here's the only place that we make FADH2 in this cycle. So that's where 2 hydrogens get pulled off. And then finally, 1 more NADH, 1 final hydrogen gets pulled off before you start the circle all the way over again. So if we were going to summarize this, how many NADHs per turn? Those are the orange ones. There's this one up here, this one at 3, and then one at 4. So I have 3 NADHs times 2, because times 2 turns. How many FADH2s? That's the green one. 1 FADH2 times 2, because I'm going to run the TCA cycle 2 times. I'm going to do this series of reactions twice. How much ATP? 1 ATP per turn, but I'm going to run it twice. How much carbon dioxide? I get 1 here and 1 here. So in this one, I get 4 of my carbons from glucose. Remember that I lost a carbon dioxide off when it came in. So there was a CO2 that came off when it was coming into the mitochondria. That's where all my carbons go, from glucose. So this is really where your carbon dioxide is made. So CO2 is made in this middle part. But do you see any oxygen used? No. So some people are like, well, why isn't this anaerobic too? So the issue is all these NADHs and FADH2s, I have to do something with them. They can't just sit there with their hydrogens. They're kind of like energized because they have that hydrogen, and they want to pass that hydrogen off, and that happens in the 3rd step. So in the 3rd step, this happens. So NADH, this is made in glycolysis and in the citric acid cycle. FADH2, made just in the citric acid cycle. It takes the hydrogen and it splits the hydrogen into a hydrogen ion and an electron. So if you remember, hydrogen is element number 1, has 1 proton and 1 electron. So really what the enzymes do is they actually split the hydrogen. The hydrogen ions get pumped out. So you'll have lots of hydrogen ions pumped out. Same thing with FADH2. Those hydrogen ions from the FADH2 get pumped out. And it is what these enzymes in the inner membrane. So remember I told you that the inner membrane is the real ruffly looking one on the mitochondria. So those enzymes take the hydrogen, split it, and push hydrogen ions out into that space, the space between the inner membrane and the outer cover. So we start developing lots and lots of hydrogen ions out here. The purple complexes are really just proteins that act as enzymes, and they pass electrons. They pass electrons along from one to the next, to the next, to the next. And that's why they call this electron transport, because it transports electrons along this membrane. And the big thing is it's going to make a high concentration of hydrogen ions outside of the membrane, low concentration of hydrogen ions in. So at the end of this, the electrons are going to be moved back into the mitochondrial space, back into that inner part. And there's a third enzyme that comes into play. They call this ATP synthase. This is your ATP generator. Generates ATP, and it uses the hydrogen ions to run it. So remember I said all these H pluses? So they show that just the outside looks really positive, but there's lots of H pluses, lots of H pluses. See all these H pluses get pumped out? So there's not very many inside, but these H pluses can actually come back into the mitochondria, going from high concentration to low concentration. Remember, that's the normal process. Things want to go from high concentration to low concentration by diffusion. But as they go through, it actually causes this to generate ATP. So one NADH can generate 2.5 ATP. So to every NADH that's made in glycolysis in the citric acid cycle can make 2.5 ATPs. FAD is a little further down the line, so FADH, it only makes about 1.5 because it doesn't use that first enzyme complex. It comes in on the second one, so a little less energy. So let's look at how many of those we made, though. So we made some ATP directly, but most of what we made happens because of NADH and FADH2. So in glycolysis, so remember that we said that this is the anaerobic step. This happens in the cytoplasm. We make 2 ATPs directly just by we put in 4, but we get 2 out. We said we make 2 NADH molecules in glycolysis. Each one can then be used to generate 2.5 ATPs in electron transport. So that would be what? 5. So glycolysis, if we can do it aerobically, we can end up with 7 ATPs in glycolysis. So then there's that step, the step to take pyruvate and move it into the mitochondria. And doing that, you make an NADH for each one of the pyruvates. So that's 2 of them times 2.5. So that would give us another 5. And then in the citric acid cycle, we made 3 and we had to do 2 turns. So because we do 2 turns of the cycle, we make twice as much. 2 GTPs become 2 ATPs directly. 6 NADHs gives us how many ATPs total? 6 times 2.5. 15. So we get 15. We have 1 FADH2, but we do 2 turns, so that's 2 of them. Times 1.5 gives us 3. So if we take glucose and we aerobically break it down, how many do we get total? 2 plus 5 plus 5 is 12. Plus 2 is 14 plus 15. 29 plus 3. 32. So see how much more energy you can make aerobically? We said we can make 2 ATPs per glucose anaerobically if we just do glycolysis. If we only did this, we're going to make 2. But if we can do it aerobically, we can make 32 ATP from one glucose molecule. So that's where the mitochondria really come into play, because all of this happens in the mitochondria. That's why they call the mitochondria the powerhouses. They're really the energy generators. So if you look at 1 C6H12O6 plus 6 O2 makes 6 CO2s plus 6 H2Os. This is like a combustion reaction, right? Because anything plus oxygen making carbon dioxide and water, so that's like burning, like you would burn wood or paper or gasoline. It's a combustion reaction. But in this catabolism, in this breakdown reaction, we use glucose. We need oxygen. So the oxygen comes into play, and I didn't actually show it to you. The oxygen comes into play at the very end of electron transport. So the oxygen combines with the hydrogens. So the hydrogens, the electrons and their hydrogen ions reform back into hydrogens, and that's where they go. So this is why it's aerobic. In order to do the last 2 steps, you have to have oxygen. If you don't have oxygen, then too much NADH and FADH2 builds up in the cell, and you can't continue to run this because there's got to be some place for the hydrogens and the electron, the hydrogen ions and electrons to go. So that's why it's an aerobic process. And look what it makes. So it makes water. So that is on the product side. So in our like summary reaction, here's the water. So that's formed in the mitochondria. In carbon dioxide, remember, carbon dioxide actually gets clipped off primarily in that citric acid cycle. So this just sort of like summarizes the process. The purple up at the top shows glycolysis. Only get 2 ATPs, get 2 NADHs. The middle part where you have the purple little circle, that's the citric acid cycle. Lots of NADHs, lots of FADH2s, and all those go into electron transport because that's where your real energy generation comes in. So that's where lots of ATP gets generated. All this in this box is all mitochondria, cytoplasm's only glycolysis. So you can use the others too. So in this one, this shows the other nutrients. So remember we said that you've got proteins, amino acids, sugars, and then the fatty acids. So we can actually shuttle them in. But notice, so this step right here, this is glycolysis, right? Taking the monosaccharide, making it to pyruvate. That's the breaking of glucose in half. And amino acids and fatty acids don't do that step. They actually go directly into the citric acid cycle. So they end up getting put into the steps just above or directly into the citric acid cycle, which is where you really get a lot more NADHs. That's really where you get a lot more of your ATP production. The challenge is with amino acids, the backbone of all those amino acids, there's a nitrogen, carbon, carbon. That's that part of the amino acids that all 20 amino acids have in common. But notice that glucose does not have nitrogen. So in order to utilize amino acids for energy, you have to take off the nitrogen. So the nitrogen has to be removed from every single amino acid. If you're going to use that amino acid for energy, you have to pull off the nitrogen. Nitrogen on its own becomes ammonia. And so ammonia is a strong base, ends up having an effect on blood pH. So you take the nitrogen and convert it to a molecule called urea. And urea is not a base. Urea is neutral. Urea actually has 3 nitrogen molecules combined together. And then what happens to that? It dissolves in water, floats around in your blood, so your cells dump it out. And where does it go? Where do you find urea?
Urine.
In urine. So that is actually your major nitrogen waste. So the more muscle metabolism, the more you use any kind of protein for energy, the more urea you're actually going to produce, and the more your kidneys have to filter it. So if people are like working out and trying to like build muscle, they usually increase their protein intake, and it's really important to make sure that you get a lot of water because you're going to have a lot of nitrogen waste. And if you don't, then your urine is going to be more concentrated, which means that your kidneys are having to work overtime trying to filter all that nitrogen waste out. So with this one, this nitrogen waste, common with high protein diets, people would be on a high protein diet, especially if they're trying to build muscle, if they're working out, then they're going to need more building blocks to build muscle. But they'll use more of those amino acids for energy too. That's going to increase their nitrogen waste, and this is why you want to increase water intake. So the other one is if you use fatty acids. So you can use glycerol, but glycerol is really small. It's just a 3-carbon unit. It's not really large. It's really those long carbon chains, the fatty acids, that you can use for energy. So when you use fatty acids for energy, they don't have the oxygen. Fatty acids are just carbon, carbon, carbon with hydrogens. Lots of carbons and hydrogens, not very much oxygen. So what do you know about calorie content of fats versus carbohydrates? Which is more? Fats versus carbohydrates. Which one has more calories per gram? Fats. Okay, fats are 9, carbohydrates are only 4. Okay, the reason that fats have more energy is because there's not a lot of oxygen in them. So they actually are going to release more energy in their complete breakdown. So in this one shows 1 fatty acid. Typical size is about 18 carbons long. This process of breaking down fatty acids which is your fats and oils, the long carbon chains in the triglycerides that got broken down. So in this, you actually just go along, and you clip off 2 carbon groups. So if you have 18 carbons, that means you can actually make 9 acetyl-CoAs. So remember the 2 carbon group that goes into the TCA cycle? You can make 9 of those. You can run the TCA cycle 9 times. Glucose, you only run it twice. So this, you have lots of TCA cycles. So from an 18 carbon fatty acid, you can run 9 citric acid cycles. Because we're going to make 9 acetyl-CoAs. Which is the piece that feeds into the citric acid cycle, that circular series of reactions. So in that, you can make more than twice the amount of energy compared to a carbohydrate. And so that's why fats have a higher calorie content. Burning fats uses almost entirely mitochondria. You don't do glycolysis, so you don't do anything in the cytoplasm. As soon as these fats come in, as they're broken down in the cell, they automatically get shuttled into the mitochondria. breakdown products, if you have a lot of acetyl-CoA, if you have a lot of those little 2-carbon units, when they break up or when they build up, they make things like hydroxybutyrate, acetoacetate, and things that they call ketones. So ketones have an acidic pH, and for diabetics, because remember the diabetics don't have insulin, so their bodies can't use glucose, can't use carbohydrates. So their body relies more on fats to break down, and they end up building up lots of these ketone molecules that can create ketoacidosis. For somebody that does low carb, high fat, kind of moderate protein, that is sort of the new kind of diet rage that they call a keto diet. That is where you're eating more fats. You still don't make the acids that a diabetic would make. And if you're not eating carbs, what's happening to your blood sugar? If you're not eating carbs, it stays the same. So that's the big difference. So somebody that's a diabetic, they tend to still eat carbs, and so they have hyperglycemia, and they're generating a lot of acids as this byproduct, and their body's breaking down a lot of protein to try and counter the fact that they're not having any carbs at all. So somebody that's actually limiting carbs on purpose does not have the hyperglycemia, so they're getting most of their energy from fats, some of it supplemented by protein. So they're actually using their mitochondria even more and actually generating more energy per gram compared to those carbohydrates. So that's what, when they talk about ketosis, it's really just how much fat burning is actually going on. It is a representative or a measurement of fat metabolism. So you may have done, if you've had AMP and you did your urinalysis, you may actually do your dipstick and check for ketones. So ketones is an indication of just how much fat you're actually burning compared to carbohydrates and proteins. But here again, we're making a lot of these ketones. And so some of them will get broken down and used by cells, but a lot of that ends up either getting exhaled, which is why people that have in ketosis may have kind of a fruity breath, like a diabetic. It smells almost like they've been chewing juicy fruit gum. It has that kind of smell to it. Have you ever smelled puppy breath? Like, have you ever smelled like a 6 to 8 week old puppy? They have like that sweet smelling breath. That's ketosis. That's because during the night or while they were sleeping, they actually did more fat burning to try and generate heat to keep themselves warm. That's that kind of smell. So that's what you would recognize on somebody's breath that's doing a lot of fat metabolism. But the other thing is also kidneys. So the kidneys are going to filter out these byproducts. So if you have a lot of protein in your diet or a lot of fat in your diet, the amount of water in your diet should be substantial compared to having a carbohydrate diet. Because when you have carbohydrates, you make carbon dioxide and water. You make water. Okay, it's one of the products in there. So it's not as in carbon dioxide, as long as your lungs work, you get rid of carbon dioxide with no problem. Your kidneys don't come into play. But when you're talking about fat metabolism or protein metabolism, if you're going to use either of those for energy, the amount of water that you need to ingest is like twice as much as what the average diet would require. It's kind of interesting though, because I've been doing some reading. They're now finally starting to really do research about like, is this beneficial? Is it good? And like my daughter's a dietitian, and she said like the hardest thing is just trying to make people actually do this. Because she's like, everybody just wants to go back and eat bread. She's like, people just want to eat the starchy foods. And all your life you've been told like high fat diets and that it's better if you have low fat diets. Like that's what, like all your life you've been told that that's the right way. But now they're actually showing that you actually have better cell repair under these conditions. And that mice that are like have gone through like these lower carb types of diets actually live longer, that actually have less disease development. Just sort of an interesting one. And I think in like maybe the next 5-10 years, you may start finally seeing the American Dietetics Association is like So slow to make any kinds of changes. But they initially discovered the keto diet for patients that have epilepsy. So they would put them on a strict, limited carbohydrate diet, less than 50 grams of carbohydrates a day. So that's like literally you eat nothing with any kind of starch. Nothing. Because you're still going to get small amounts from various things that you do eat, but you don't eat anything with any kind of starch. And they found that that actually slowed, like epileptics that were not responding to medication, that would actually limit their epileptic seizures. Still don't I've never really read why they think that happens, but that's how this diet actually originated, is because of that. All right, so on this, that finishes these. Like I said, just like the summary with each one of them, that's really the ones to focus, like where is carbon dioxide made? Why do you need oxygen? What is NADH? What is FADH2? Like in general, what happens in each of the cycle, in each of the steps of glycolysis versus the citric acid cycle versus electron transport. Just try and kind of keep it as simple as this compared to like the material in the book. And I really don't have anything about like the steps involved with amino acid metabolism or fat metabolism, other than the fact that like if you ingest a lot of proteins, you got to get rid of the nitrogen. If you ingest a lot of fat, you're going to have some ketones that you have to deal with. Those are pretty much the only big ones that are different. All right, I will have this posted